Good morning, everybody. It's a real pleasure and honor to be here with all of you, and especially Clark and his family. To understand Clark's legacy is that you have to go back to the 1970s. So at that time, the boundaries of a chemical engineer was basically you were a traditional engineer or you were a biologist. There was no difference. There was a wide difference between those two areas. So in 1976, I had the distinct honor of being one of the first 3 students doing interdisciplinary thesis. And that was truly a multidisciplinary thesis. That was a test case that Clark spearheaded, and he was the architect of that. Let's see. Our goal was to design a revolutionary batch reactor that used enzymes in an aqueous medium to produce the adenosine triphosphate, ATP. At that time, we wanted ATP to was only made by biological process like fermentation, which is a slow process. So we wanted to engineer that. I remember the late nights in the lab, deconstruction of the Krebs cycle, as it was a refinery process. Clark, your rigor of legendary approach here was that you couldn't just accept the results just because it worked. You demanded a full understanding of the fundamental transport and the kinetics. So when we finally succeeded making the so-called ATP production system, the enzymatic reactor, the impact was immediate. We had it was an academic exercise that resulted in a commercial design. The major pharmaceutical companies ended up getting patents and license to that, and that changed some of the production methods using enzymes to produce products in an aqueous media. But for me, the product wasn't just the ATP. It was really the realization installed by Clark that the tools of chemical engineering seemed universal. He taught us that the molecule doesn't care if it's made in a petrochemical plant or in a human system. So he said, basically, follow the laws of the mass transfer, and if you master that, you can do almost anything. I ended up doing most of my career in the industry, and so to give you a little glimpse of that, I worked at Procter & Gamble. That's my first job after coming out of my thesis. And I worked on a product called Bounty, and many of the paper products at P&G. And one of the products was Bounty. And the Bounty, anytime you use Bounty, they had this so-called promotional activity that says quicker, picker, upper. So anytime you use Bounty, just think about Clark. He contributed to that. So armed with this multidisciplinary approach, I ended up doing quite a few tasks within the corporate giants like DuPont and P&G. And so people at that time, they were looking for specialists in their field, but it turned out that I was a generalist, as generated by my interaction with Clark, But I was journalist with specialized precision. Yeah, let me mention the DuPont. The DuPont at that time was a huge, probably the largest chemical company at that time. And I worked on Kevlar. Kevlar was one of the major projects, especially for armor applications, military applications. And we ended up creating artificial intelligence system, we called it at that time, that we could actually do production planning and scheduling, taking a look at the supplies and demand, and we could match that across the whole business. And that was a major product opportunity that we ended up being able to do that, and we got some recognition in the industry that this was the first artificial intelligence application. So armed with this the capacity to bridge the so-called separate worlds, we ended up doing some joint ventures between DuPont and Conagra. And these efforts ended up, I need to get some water here. Let me see. Thank you very much. Thank you. Yeah, thank you. Let me go back a little bit. Well, whether it was, I was in the R&D and managing large manufacturing opportunities, and I constantly applied Colton's approach in deconstruction, and I found myself leading teams where I had to speak the language of both as a chemist, as an engineer, and also as a business executive simultaneously. So Clark, you taught us how to do that, and we had to basically this multidisciplinary approach has ended up being quite a buzzword out in the field, but really it's the key to having the ability to carry out science. So this complexity of global manufacturing with the same curiosity that we had in the lab in '76. So with this approach of having the ability to take products that were sitting in these large companies, not unused, and we ended up creating joint ventures with a company called Conagra, and we had multiple products coming out of that. And one of the crown jewels of that was a product that is used passive immunization. And this product we used a polyvalent vaccine to generate a product that contained ingredients, the so-called protective antibodies that cover most of the aggressive diseases that afflict humans. And this polyvalent vaccine contained 26 different antigens, and we were able to create antibodies against each one of those. So we didn't stop with the antibodies, but we ended up generating a matrix of So we ended up generating a matrix of products that were anti-inflammatory, so we could combine the two technologies. And this product has proven to be invaluable as a tool for cancer patients undergoing chemotherapy. So Clark, this was an important combined, this project combined both immunology, large-scale agricultural infrastructure, and advanced engineering. It was an ultimate validation of what you taught us to do. That engineer can look at an egg or a vaccine and see the beautiful, complex transport and production system. So I ended up going into the so-called, really carrying out the philosophy to deal with advanced diseases like controlling the stage 4 metastatic diseases. And so when you're dealing with advanced life-threatening illness, the system you're engineering at the human response, it's the ultimate multidisciplinary approach that you need to take a look at, and it requires an understanding of molecular biology, kinetics of the cell-to-cell interaction, and the delivery of revolutionary therapy. So in these moments of high stakes research, I would then think back to our discussion of the Institute. I would ask myself the question Clark always asked us to do. He said, look at the whole system, but don't look at the things that you would like to see. So treating cancer at the most advanced stage is about managing the most complex reactor in existence, the human body. So Clark, the foundation you gave me allowed me to approach these projects from that aspect, and ended up being quite proud of being an MIT engineer. So Clark, you have spent 50 years now in redefining what it means to be a chemical engineer. You took the discipline rooted in basically just steel and steam and moved it to the realm of DNA and immunotherapy. Your legacy isn't just in the published papers or patents, but it is the fact. It's the fact today that multidisciplinary approach is the key to science. You were the pioneer you saw the coming of this multidisciplinary approach better than anybody else. So the rest of the world is catching up to what you've done. To those who sat in your office and felt the Colton heat of a rigorous critique, well, we thank you for that. It challenged us to take the most meaningful actions. You didn't just teach us how to do the work, but you taught us how to think. You gave us the tools to synthesize the energy of life, and eventually tools to save it. So to the architect of modern engineer, the master of the multidisciplinary approach, and my mentor, Clark Colton, we wish you all the best, and we are looking forward to new discoveries.
Thank you, Clark. Thank you, Kathleen, for giving me an opportunity to speak at this great event, and Clark, to express my deep gratitude for your unwavering support during my SC thesis, to have enabled a crazy French student to develop something completely unexpected and unforeseen, namely high gradient magnetic filtration to move polyacrylamide beads with immobilized enzymes through various reactors and purification steps. As you know, the idea of using magnetic particles or beads for biological applications, biotech application, has made its way. What I've done since, been mostly a serial entrepreneur, started a series of companies, 10 made it on NASDAQ, one on the London Stock Exchange, one on the Swiss Stock Exchange, managed 2 funds, the Harvard Medical Venture Fund, and then a large fund I created with Hoffmann La Roche on UBS in Switzerland. And now I'm very involved in my probably most exciting creation. 2 years ago, we started a company to come up with a solution to current asymmetric drone wars. We invented a low-cost solution to neutralize $20,000 Shide drones for only $1,000 to $4,000. More importantly, neutralize swarms of Shide drones, up to thousands or more Shide drones, by swarms of our own drones. And the crux of our system has been invented by a genial French mathematician, and we basically have embarked distributed intelligence without human operators, without central command, which enables swarms of drones to operate like ant colonies with redundant, high redundancies, high adaptive capability, following only one objective, not vulnerable to jamming, no use of GPS, and cannot be misused, unlike the worries about cloud. So we're meeting a lot of success. We've been selected by NATO, and now we are supported by a very impressive group of top US political figures and military advisors. We are bringing the company to the US, and we want to do a direct listing towards the end of this year. So anyway, I wish you and friends, in the audience, particularly Bob Langer, whom we worked on the same project many years ago, a great event. And should you come to France or Switzerland, I would love to invite you. So have very nice exchanges, and congratulations.
Dear Clark and Clark students and friends, my name is José Costa. Greetings from my hometown of Valencia, Spain, a city that, according to Forbes magazine, has been ranked as the best city in the world to live in. At the suggestion of the symposium organizers, since I was unable to be present in person due to other conflicts, I was invited to share with all of you my professional journey that I will name From Science to Diplomacy. So let's go back in time to the first week of September of 1980, when, after 6 years of graduate school at MIT, I managed to defend my thesis, Kinetic and Reactor Studies of the Enzymatic Regeneration of ATP. I mentioned the title of the thesis to demonstrate my interest in biotechnology. I have to mention that while working on the thesis, I took courses in biochemistry, biochemical engineering, microbiology, etc. So I think I was well trained in the field. After defending the thesis, I returned to Valencia, where I got a teaching position at the Polytechnical University of Valencia. I was 2 years in the job when I was informed that the United Nations Industrial Development Organization, UNIDO, was trying to set up an international center for genetic engineering and biotechnology, ICGEB, and needed experts in the field. I applied and got a position in UNIDO to help in the implementation of this novel initiative. This required a new international move with the family from Valencia, Spain, to Vienna, Austria. While at UNIDO, I had to tap into the know-how of international experts in the field in order to elaborate a work program for the center, as well as, and this was the most difficult part, evaluate the proposals made by member states to host the mentioned center. We had to visit scientific facilities of candidate countries and evaluate if they could attract first-class scientists to work at the center. It seems that my life was settled since my wife, who also has a PhD in chemistry, was working at UNIDO when, 3 years into working at UNIDO, I received an offer from the regional government of Valencia to become head of scientific policy. The job entitled Evaluating and, when appropriate, funding scientific research proposals. This was meant to climb a step in my professional ladder at the price of detaching myself from biotechnology. It also meant another international move from Vienna back to Valencia. So pack again and move to Valencia. By 1987, I was managing science and technology and had conducted no research whatsoever. And it was time for me to take a crucial career decision. Either return to scientific research or focus my professional career to science and technology management. I decided to pursue the latter. Did I have all the tools to become a good manager? Since this was not the case, I applied and got a Fulbright fellowship to do a mid-career master in public administration at a school up the river from MIT, the Harvard Kennedy School. A new overseas move, and through taking the family, now of 6 members, from Valencia back to Boston. After graduating in May '88, I spent the last 2 months of the fellowship visiting and getting acquainted with programs and methodology of funding research institutions like NSF, NIH, NIST, USDA. In the meantime, Spain has made its accession to the European Union, and the Commission was looking for Spaniards to work at the institution. I applied and got a position there. This resulted in the 6th international transfer, this time from Boston to Brussels, Belgium. While in Brussels, I was managing research programs of cooperative nature between industry and academic institutions in the field of microelectronics. My expectation was that this would be a permanent position where I would spend the rest of my career. Wrong. After working at the Commission in Brussels for 5 years, the Commission announced opening of the post of Science and Technology Counselor at its embassy in Washington. How could not apply for the post? I was a product of the American educational system and knew the functioning of the American institution. So yes, I applied and got the post. So, next transfer from Brussels to Washington, D.C. During my duty time in D.C., we negotiated a science and technology agreement between the US government and the European Commission. So institutions from either side of the Atlantic could participate in research program and get the potential benefit of research results. After the 4-year tour of duty in DC, it was time to transfer to another embassy. This time, the destination was the embassy of the European Union to the international organization in Vienna. This encompassed both the UN system based in Vienna, such as UNIDO, where I had worked in the past, as well as the International Atomic Energy Agency. So while my job in Washington was focused on science and technology policy, now in Vienna, the emphasis was more on diplomacy than in science and technology. After 9 years of duty in the Foreign Service of the EC, I returned to Brussels. I went back not to the Department of Research, where I began my engagement in the Commission, but to the Department of Foreign Relations. There, I covered all kinds of diplomatic relations of the EC with countries like Switzerland or San Marino. In 2004, when the European Union underwent its biggest enlargement in terms of countries, the Commission needed to make room among its staff for nationals of those countries if offered the possibility of early retirement to those who fulfill certain requirements. Since I was one of those, I applied and got my retirement in December 2004. So over 21 years ago, I returned definitely to my hometown of Valencia. You could agree that I have quite an interesting professional life, that I could not have it without my educational background, in particular, my education from MIT. My good professor and friend, Clark Colton, helped instill in me an open and curious mind. My professional journey in science diplomacy has catalyzed the use of science to foster connection across countries and regions. Interesting, rewarding, but what about the family? I was extremely lucky. They were able to adapt quite well to this non-stop moving from country to country. And like Clark instilled in me a passion for science, engineering, and research, I'm happy to report that I also instilled an interest in and appreciation for the science to my family. 2 of my 4 children went off to study engineering themselves at the Imperial College in London. And I'm happy to report that my oldest grandchild will also continue to pursue engineering at university next year. As we celebrate the professional accomplishment of Professor Clark Colton, do not forget the power of science to bridge differences in an otherwise unstable world. Enjoy the symposium.
So this is a very special event for me. I have been a friend of Clark Colton for over 40 years, and we have interacted both scientifically and as a friendship. And of course, if you look at my title, you'll see we play tennis together. I'm not going to really talk about who I am and what I've done and how, if I've accomplished anything, but I do have some history interactions with Clark, and I would like to speak to Clark and his family, especially his wife, Ellen, at the back. And this won't take 15 minutes. So I have spent 3 sabbatical leaves at MIT, 1 at Caltech, 1 at Berkeley, and 1 at Yale. So it's a long career. The career is extended every 7 years. And my visits to MIT have always been warm and friendly, especially with my friend Clark. I've been hosted by Bob Langer twice and Ronnie Probstein once, and I've made good friends in the chemical engineering department. I'm from RPI down the road. I believe it's the oldest chemical engineering, if not chemical engineering, it's the oldest university in the English-speaking world, technical university. We celebrated 200 years last year or the year before. So what I'd like to tell you about is that it's been an absolute pleasure to know Clark, and he's a very unusual person, as many of you know, or haven't said so. I enjoyed George's talk and, of course, all the other talks this morning, lots of luminaries. And I should say, Clark, you should be very proud. You've had some wonderful success and people that have emanated from your group. I would like to say a few words about, I have two short stories, actually. One of them was when I was on sabbatical here, Clark came to see me at my office, and he said to me, George, you know, my wife's a professional actress. Would you like to go and see a play? So I said, yeah, I'd love to. When is it? He said, tonight. I said, okay. So we went over, he drove me over, we went there, we had our seats already. I thought that was funny. The seats were already prepared. Everything was set. Then the play took place. And I'm not sure if Ellen remembers, but it was a play in which somebody from the audience had to come up to the stage and answer some questions. Unfortunately, I wasn't listening very clearly. And guess who was set up? Me. I went up and stood there like a complete idiot. I had no idea, and they asked me questions about the play and everything else. And I thought, my God, this was a setup. And Clark laughed and thought it was terrific. And then I said to him, you know, I have to drive home, but it's difficult in Boston because the actual signs don't tell you where to go. If you live in California, every sign counts. Here, you got to, so Clark says to me, George, you got to first know where you're going. Then you can use the sign. I said, terrific. That was one story. I should tell you, technically, I've also had interactions with Clark through a beautiful PhD thesis that I read the whole thesis by Andrew Zidney, who's going to talk this afternoon. And Andrew has been a friend of mine, and we have taught short courses to industry on membranes and filtering proteins. As you heard, one of the number 6 topic was proteins and filtration with membranes. And actually, we just published a paper this year, an unusual paper, where we discovered that proteins actually, if you have hydrogen bondings, they will hydrogen bond to a surface. And so that was the first time that I ever saw anybody mention that hydrogen bonding could be very important. The other thing is that for 20 years or 30 years, I worked on proteins. And then I decided to change to RNA completely. In fact, during the pandemic, I thought to myself, at my young age, why don't I just change direction? And so the whole group changed, and so we've really working on some serious problems on how to purify RNA, not only for vaccines, which all of you know about. Some of you get bad responses, and some of you get good responses, like me. Unfortunately, my wife is an RNA expert, and she gets a terrible response from the RNA booster. And the reason that you'd get that is because there's small amounts of impurities there. Those impurities don't kill you. They don't cause you to go to hospital, but they do cause you sort of immediate fever and shaking, and that's what I see every 6 months in my house from my wife's reaction. So it's a challenging problem, and since RNA is going to, in my view, much easier to purify, much easier to make than proteins, it's going to eventually, over 20 years, replace proteins by placing it into your body so that you make the proteins and you don't have to synthesize them outside. So that's what we're doing now. And I know Andrew is also working on DNA and RNA concentrations and purification. My last story is about tennis. So Clark's son asked me above, are you a good tennis player? I said, no, I'm a terrible tennis player. And so every meeting we would go to, Clark and I, whether it was the Engineering Foundation meeting, remember we went to some in South Carolina, or we've been to others. We've been to Whistler. We went to one in Whistler. We played a tennis game against 2 Danish players. And you and I we had this record, and you said to me, George, stop the game in the middle. So I told the 2 Danish players on the other side, would you just hold on a minute? My partner wants to ask me an important question. So I walked over to you and I said, Clark, what's going on? You said, George, we have never beaten anybody in all our years. This is the first time we're leading. Should we tell them? So I said, yes, let's tell them. So we told them, and of course, you know what happened. They beat us. I met that guy 2 weeks ago. His name is Arne Staby, and he was a famous guy from Denmark, worked for Novo Nordisk. I was at a recovery conference 2 weeks ago. I asked him if he remembered you, and he said he remembered the tennis game. So you're a memorable person, both for tennis and for all your contributions to all your students. And I just want to thank everybody for making this such a love fest. It's a wonderful day to be here with all of you. And Clark, thank you for being my friend for so long. I want to just thank you. You're a wonderful guy.
Good afternoon, everyone. It's my pleasure to welcome you to the second half of this symposium celebrating the legacy and career of Clark and his students. And so my job for you this afternoon is to give a little bit of Clark's backstory. I know you heard some of it this morning. And talk about his contributions to the department. My name is Chris Prather, by the way. I didn't introduce myself. I'm the department head for chemical engineering and also an undergraduate alum, class of 1994. Clark has been a member of our faculty for 55 years. And during that time, he has been valued as an instructor, as a colleague, and as a collaborator, contributing to both our research and teaching communities. He's a pioneer in the field of biomedical engineering, and he helped to revitalize our undergraduate lab classes. Now, I also shared an office suite with Clark, and I'll talk about that tonight at dinner because I don't have enough time to do that and do the. Clark is trying to remember those years. Just a bit of background information. Clark received his bachelor's degree from Cornell in 1964 and his PhD from MIT in chemical engineering in 1969. And I do remember seeing Clark get his red jacket, which is given to all alums of MIT on the occasion of their 50th reunion just a few years ago. Inspired by Ed Merrill and Ken Smith, whom you'll hear from this evening, his research focused on biomedical applications of chemical engineering, specifically the first study to understand mass transfer in hemodialysis, or the artificial kidney. Post-graduation, he became a professor here in the department and pursued a wide variety of research interests, including hemofiltration, which was around a better artificial kidney, ultrafiltration of protein solutions, blood microfiltration for plasmapheresis, oxygen transport in blood, enzyme engineering and transport of lipoproteins across endothelium into blood vessel walls with applications to atherosclerosis. And you heard from many of the students who worked with Clark in these areas this morning. Of course, you'll hear from more this afternoon. His research related to diabetes spanned his entire career, beginning with development of an implantable electrochemical glucose sensor to be coupled with an insulin pump for glucose control, and then a hybrid artificial pancreas comprised of islet cells encapsulated in a polymeric device. He pioneered the study of the role of oxygen and the performance of these devices and their effect on islet insulin secretion. And he was the first academic chemical engineer at a major research university who spent his entire research career mainly on biomedical subjects. And as a result of that, his students pioneered many new areas of biomedical research and went into highly diverse careers not previously populated by chemical engineers. And I have to say that from the very beginning of talking about this symposium, Clark's focus was on the students, that he really did value the time that he spent with all of you and your contributions working with him together to bring new insights and to apply chemical engineering fundamentals to new problems in new ways. So those are all things that you could have read about and maybe a Google Scholar search. But most of what I want to talk about is Clark's role in the department. He served as acting department head for 1 year in 1977 and 78. And most people who were going to be an acting department head would just sit around and sort of figure out, how to pass the time until somebody came in and took care of all the stuff that he needed to be taken care of. Clark didn't do that. He actually appointed the first graduate admissions committee for our department. And all of you who are academics know how critical it is to be able to recruit talented students into our universities to continue the work that we're doing. And Clark set up the basic structure of that and chaired that committee for 10 years. We still use the basic framework of the system that was set in place by Clark today. And our graduate recruiting process, of course, is an all-department effort that continues to impress students with the time that we as faculty spend with them and understanding what their interests are and in trying to make it clear that they understand the role that we play in supporting their educational endeavors when they arrive. In 1988, he chaired the celebration of 100 years of Course 10, the Centennial of Chemical Engineering Education. This was an event that brought leading scholars in ChemE to a retreat off campus to discuss the future of chemical engineering, followed by a convocation that filled Kresge Auditorium with alumni and friends of the department. And those of you who attended that symposium will remember it, because I still hear from people who attended that symposium all those years ago about what a remarkable event it was to celebrate the launch of a discipline. We had the first chemical engineering department in the country here with the first students admitted in 1888. And it really did establish MIT as a place that was thinking not just about what chemical engineering had been and what it would become. And Clark's role in managing that celebration is actually the inspiration for a celebration we will have in 4 years to celebrate our 110th anniversary of the program or our super centennial. By the way, if you're wondering, you can find the videos of that event on somewhere in the ether on the internet. I actually stumbled upon it by accident about 8 years ago. And again, it really is a remarkable way to celebrate the discipline and Clark's vision in terms of being able to bring together leading scholars to emphasize the importance of our field. Clark has left an indelible mark on both undergraduate and graduate education in the department. He had the unique perspective of being both a student of and then a professor for Course 10.56, Chemical Engineering in Medicine. So again, thinking about how do we take these fundamentals of chemical engineering and apply them in new ways. And he taught several bioengineering courses. In the 90s, Clark became notorious, infamous, for revitalizing what we call Project Lab, Course 1026, 27, and 1029, and brought in industry partners to provide students with the opportunity to solve real-world chemical engineering problems while learning to communicate professionally and navigate team dynamics. I was actually an undergraduate in the 90s. I did not take the class, don't tell Clark, but I remember M&Ms. Clark, you remember the M&Ms? There were, I don't know how many 55-gallon drums of M&Ms because one of the projects was about coating and the candy coating that goes onto M&Ms. And I had upper class friends who would come back to the dorm with lots and lots of white M&Ms because after a certain point, you just had to give them away and eat them as they were. The project, team management and the project management aspect of this project lab was something that Clark brought to the class and continues to be a unique aspect of that class, and something that students don't get in other places. They see it as differentiating as a part of their education. I said don't tell Clark I didn't take it because Clark firmly believes, and if I gave him the mic when I was done, which I'm not going to do, if I gave him the mic when I was done, he would start talking about how every student should not be allowed, no student should be allowed to graduate without having gone through Project Lab. Do I have that right? He's nodding. And it is a class that continues to attract a lot of students and really allows them to explore their independence in using the fundamentals, again, of chemical engineering to solve new problems where they don't already know the answer. And this is, of course, the foundation of a graduate education, but to have the opportunity to teach students in this way with a focus on undergraduate education really is a distinguishing feature of the course, and we owe that structure of the course and the legacy it's been able to leave to Clark and his work there. Clark has gotten, as you might imagine, a number of awards. These include the Camille and Henry Dreyfuss Foundation Teacher Scholar Award, the Alan Cobord Award from AICHE, the Curtis W. McGraw Research Award from ASEE, the Gambro A.B. Award from the International Society of Blood Purification, a Lifetime Contribution Award on Bioartificial Organs from the Engineering Foundation, and then a Division 15, which is the Food, Pharmaceutical, and Bioengineering Division Award from AICHE, and he also was included in the 100 Chemical Engineers of the Modern Era. He's a founding fellow of the American Institute of Medical and Biological Engineering, and he was also an elected fellow of the American Association for the Advancement of Science and the Biomedical Engineering Society. Adrian has raised the sign that says, "Warning, you have 1 minute left," and the warning part is very terrifying, even as a department head. So I will wrap this up by just saying that Clark's imprint on the department is erasable. It will be here for a lifetime, and we really genuinely appreciate the commitment, Clark, that you have made to this department, and in particular to the education of our students over the last several decades. And so I will end just by saying thank you.